So autism is a spectrum of disorders. This is taken straight from the DSM-5 manual, the diagnostic manual, that says that autism is a persistent deficits in both social communication and social interaction across multiple contexts. So you're not just at home or at work, but at school and out in the community. And it's also accompanied by restricted or repetitive patterns of behavior, or very restricted or repetitive interests or activities. And symptoms must be present in the early developmental period of a child's life. The symptoms cause clinically significant impairment in social, occupational, and other important areas of functioning. And these symptoms or disturbances are not better explained by intellectual disability or global developmental delay. So that's the definition of autism. What it is, it's really a spectrum of disorders that range from very mild to very severe or profound impairments. I love this picture because it really, in a very playful way, demonstrates kind of what the autism spectrum or the autisms are. We recognize all of these as animals, but they're all completely different sizes, shapes, colors, affects, and they manifest out in the world in a very different way. This drawing actually was done by my nephew, who is autistic, and he is now about 35 years old. He drew this picture when he was maybe in his late teens, but he's been drawing these animals since he was a very young child. And we were together on a vacation that year, and I asked him to draw me a picture of his animals. I just love his animals. And he said, okay, I'm going to draw 77. And he sat down in the morning and he started from the beginning and worked his way through about here. And then it was time to go to the beach, and we were all saying, okay, let's go to the beach. And he hadn't finished all of his animals, and so he became very agitated and really needed to just stick to this and continue drawing. So I tell that little anecdotal story because it sort of characterizes a lot of features that some people with autistic people have, this rigidity to and adherence to routine, and can be very disturbed if that routine is disturbed. And this sort of persistence and repetitive, these, drawing these pictures and these animals in this way, for year after year after year, even into his young adult life. Okay, so we know that autism has prenatal origins, and we know this from a variety of different disciplines. This is research showing that from very early on, the neuroanatomical studies looking at brains of people who had passed away and who had autism, they found aberrations or malformations or differences in many different brain regions. And because of what we know about brain development and the timing of brain development, we could decipher that these insults to the brain, or the brain development, the dysregulated brain development was occurring prior to 30 weeks gestation. So early on. We also know that from early studies, this is one of the first sort of epidemiologic studies of autism, linking autism to congenital rubella. And this was the case that women who had rubella during pregnancy and delivered children, those children had much higher rates of autism than what you would expect. There's also evidence early on from studies of valproic acid, that drugs that people took for controlling seizures and took during pregnancy, and some of those children were later diagnosed with autism and at much higher rates than expected. And there were other medications used during pregnancy to control nausea that also have been linked to increased rates of autism. And more recently, this is research I've been involved with, and many others have also, looking at antidepressant use during pregnancy, and specifically SSRIs. And there's now a large body of evidence linking SSRIs to increased risk of autism, but it's still not clear if there's an independent effect of the antidepressant, independent from the indication for the drug, which would be maternal depression or anxiety or some other mental health issues. So that's still an active area of research. Nevertheless, we're talking about prenatal exposures. And then finally, also from a lot of early studies looked at obstetric complications. This could be anything from delivery complications to such as a C-section or breech delivery, to complications, pregnancy complications earlier on in gestation, preeclampsia, hypertension, many different forms of pregnancy complications. And as when you take them together and create an index of obstetric complications, that has repeatedly been linked to increased risk for autism spectrum disorders. So from many different points of view, there is strong evidence that autism has prenatal origins. We also know, though, that there's decades of research on genetics and autism, and we know that from twin studies, from family studies, from association studies, that autism has a strong genetic etiology. So the current belief and dogma is that like many other complex disorders, autism spectrum disorders arise from a combination of genetic and environmental factors, and that there are critical exposure windows in development, and they're very early on, prenatal as well as early postnatal possibly, and that the different clinical subgroups likely have different risk factor profiles. So one thing I talked about already is the range of severity from mild to severe in terms of the core symptomatology of autism. There are also many different co-occurring conditions with autism, both physical and mental health conditions that are more prevalent in individuals with autism than the general population. And we're not exactly sure that the reason why that is also multifactorial, and there's a whole nother strain of research that I'm involved with, looking at those associations with health, with physical and mental health conditions, and the reasons why those may co-occur at higher rates in autistic people, which I'm not going to go into today. So one of the environmental, so-called environmental factors that has been shown to be associated with increased risk of autism is maternal infection during pregnancy. And I'm just flashing up here many studies that have been published looking at infection during pregnancy from different angles. And as a body of literature, there's pretty strong evidence that there is some association between maternal infection and pregnancy, but there's a lot of unanswered questions still, and some of which I've listed here, which is, which infectious agent is it, or are they, that are most associated with increasing risk for autism? There's research that has looked at influenza, for example, and found increased risk associated with influenza or other viral infections. I talked about rubella. CMV, there's several different specific viral infections that have been associated with increased risk in epidemiologic studies. Also bacterial infection. Some research suggests that it's when you look at women who are hospitalized for infection, that it's actually bacterial infections that are related to the increased risk. There's also big questions about the timing of exposure. during pregnancy? Is the association related to infection in the first trimester or later on in pregnancy? And there's various, the findings are variable across time period. And then questions about, well, what is the actual risk factor? Is it the infectious agent per se or the immune response? And finally, maybe there's common underlying genetic susceptibility to women who are getting infected during pregnancy. Maybe are genetically have a genetic susceptibility for some reason to infection that's also related to a genetic susceptibility to having an offspring with autism. So lots of unanswered questions, like many fields of research. And I'm going to be focusing a lot of the rest of what I've talked about in terms of this infectious agent versus immune response. One of the studies that I recently was involved with, this is these are data from to answer this, to try to get at this question of the infectious agent versus the immune response to the infection. These are data from the Study to Explore Early Development. This is a large study funded by the CDC that's been still ongoing. And it's a large case control study that was done in originally 6 sites across the country, California, Northern California being one, in which I led that effort in California. We included the other sites were in Maryland and Philadelphia and Pennsylvania and Georgia, Colorado, North Carolina, and now a few other sites have joined. And these are data from the first wave of the SEED study that combined data from all of these different 6 locations. And where we looked at infection and all different types of infection, viral, bacterial, sort of coded it by organism and also by organ system. And then we're able to look at fever accompanying the infection or not. And what we found overall, we found no association with infection per se and in any trimester, but we did find this significant 2-fold increased risk associated with second trimester infection that was accompanied by fever. And this has been reported by at least 2 or 3 other groups using different data sets. This finding of second trimester fever increasing risk for autism. So our conclusion from this study was that there's sort of a general it's the immune response to an infection and not a particular infection per se that is elevating risk. So imagining that the immune system during pregnancy can change the way a child's brain develops. What if we could predict which children would be affected by a neurodevelopmental disorder by being able to do perhaps prenatal screening or newborn screening to identify biomarkers and maybe immune system biomarkers that would be highly related to subsequent diagnosis of autism in an offspring. That would be very helpful so that we could know very early on which kids might be on a trajectory for a neurodevelopmental disorder, and maybe specifically autism, and introduce interventions very early on that could prevent some of the morbidity associated with autism. So just to give you some, real broad brushstrokes background in immune system response, I'm going to be talking about cytokines and chemokines. These are master regulators of the immune response. Cytokines are small molecules that tell other immune cells what to do, when to become activated, when to reduce activity. And chemokines tell cells where to go, like migrating to a site of infection. And here's a sort of a very simple diagram or model of this maternal immune activation model, which we think is what's happening when someone gets an infection during pregnancy, and why it's may impact neurodevelopment. So we have these little viral particles, let's say. A mother becomes infected, she has a fever response. There's impaired immune regulation, so that there's reduced ability to control the inflammation. You have this increased inflammatory cytokine production, and here's some cytokines being produced here. They get into the maternal circulation. We have this sort of generalized maternal inflammation. This inflammation can alter the placental function, and there's placental transfer of cytokines to the fetus. The developing blood-brain barrier actually does allow access of some of these molecules to the brain, unlike what we used to think, that the blood-brain barrier was very impervious to anything. We do know that some of these molecules, not all, but some do transfer and can impact, get to the fetal brain and to the CNS. And then the inflammation can alter the CNS development. We know, so kind of showing in a different way, so that here's this prenatal environment, that's this maternal fetal immune environment, which can be seen as the risk factor. And there's very big interactions between the immune system and immune molecules and the nervous system. That the immune signaling molecules really play critical roles in all stages of fetal brain development. And when you have changes in the maternal fetal immune environment, you can have major disruptions in this very finely orchestrated events that are neurodevelopment. And you can see the different processes happening across all periods of gestation, where early on you've got neurogenesis and gliogenesis happening. In the second trimester and third, you've got neural migration and apoptosis and synaptogenesis. These are all very, very critically important processes for normal brain development. And so if any of this changes, this immune environment is changing, it can, while the fetus is developing, it can impact and possibly with adverse consequences to fetal brain development. But we know that the majority of women who are exposed to immune challenges during pregnancy give birth to neurotypical offspring. So the challenge is, it's not enough to know that someone has an infection or someone had an exuberant immune response to be able to predict what fetuses are going to go on to perhaps be diagnosed with a neurodevelopmental disorder. There's a lot of other factors at play. Genetic susceptibility is one of them, both the maternal genetics and the child's genetics. The timing of the insult, the timing of the immune challenge, the intensity of their immune response, and then additional prenatal and postnatal events that can all add in to ultimately leading to adverse neurodevelopmental outcome. So we've been interested in trying to disentangle a lot of these factors. And so the first study that really attempted to look at these factors in this sort of multifactorial way was the Early Markers for Autism study, which is affectionately called EMA. And EMA actually means mother in Hebrew. So I especially like this acronym for this study. We're studying mothers and their children. And we're investigating early biologic markers of susceptibility and exposure during critical periods of fetal brain development. So and the whole purpose of this study is to determine contributions from immunologic and genetic factors, and also environmental exposures, and the interplay between genes and environment, and in the etiology of autism spectrum disorders. The study was designed as a population-based case control study. of mother-baby pairs, taking advantage of some resources that we have in California that I think are particularly unique and are available for researchers to use. And these are these prospectively collected specimen banks, both maternal pregnancy specimens and newborn specimens that are the newborn specimens, as many of you know, are collected on every single baby and for newborn screening of many different diseases. And they're banked in huge kind of Costco style warehouses, freezers, and they can be used for research with permission from the state and all the appropriate IRB approvals. In addition to the newborn specimens, there's a bank of maternal second trimester blood specimens, and this is from the AFP, alpha fetoprotein screening program. These were leftover samples that were left over after the prenatal screening, and also banked in certain years in certain counties in California. So we took advantage of these two specimen banks and a third resource, which is the California Department of Developmental Services, the DDS system, which provides services to both children and adults with developmental disabilities statewide. And so we were able to identify children who were in that state service system receiving for services for either autism spectrum disorder or another developmental disorder, and then link those children to their mothers in the areas where both the maternal specimen bank and the newborn specimens existed. And so for this study, it was San Diego County, Orange County, and Imperial County. And then identify those women for whom we could, and children, the mom-baby pairs for whom we had both a maternal specimen and a baby specimen. And then we sampled population controls from children who were not receiving services and never did in that state service agency, and who also had a maternal and a newborn specimen available. So it's sort of a pro, I don't know what you call this exactly, prospective case control or retrospective cohort study, where the specimens were collected prospectively, but then we went, we started from the outcome and then worked backwards to identify our study population. And so this has been an incredible study. It's been, we've had a couple different phases. The funding has been over for many years, but we've continued to meet as a group monthly and analyze the data and write papers. And in fact, there's a review paper coming out very shortly in the journal Molecular Autism that is a summary of the work to date, the first 20 plus papers that we published from the study and describing everything. So what did we do? We had maternal specimens from blood specimens from mid-pregnancy and newborn specimens, and we were able to interrogate those biologic specimens for a variety of immune markers, environmental exposure, environmental exposure markers, and then also genetics in both moms and babies. And again, the main outcome was ASD with autism, but we also had this comparison group of children with other developmental disorders, and then a control population. And I'm going to show you some of the findings relating to the immune system, the maternal immune system. So one of the first papers we published was looking at autoantibodies, maternal autoantibodies to fetal brain proteins, and found that it basically replicated work that had been done by Judy Vanderwater and colleagues at UC Davis, where they had looked at postnatal maternal samples and found a specific pattern of band pattern on these plots that distinguished children with autism from children without autism. So these are maternal, this is maternal antibody patterns, and it was sort of at the 39 kilodalton, 73 kilodalton. It was actually this sort of combination. And this A column is a child with autism, or is a mother of a child with autism, and then B and C are references. And you can see this is a different pattern. And this was found in much more often in the mothers of children with autism than the control mothers. So that's one type of immune system finding that was very interesting. The other was looking at maternal cytokine profile, and we found significant differences between in three these are pro-inflammatory cytokines, interferon gamma, IL-4, and IL-5, that were elevated. Levels were elevated in the mothers of children with autism compared to the mothers of the general population controls. And this is after controlling for lots of different covariates. Then in the second phase of EMA, where we had a much larger sample size, we were able to look at phenotypic subgroups of autism, as well as just autism overall. And what's shown here is this kind of a complicated slide. This is down here are the different types of cytokines and chemokines broken into these different groups of sort of activity. And across the top here are the different groups of the phenotypic groups of our children. So this is ASD with intellectual disability versus the population controls. This is ASD without intellectual disability versus population controls, and so on. And what we find here is that when it was really this group, the ASD, I guess you can't see can you see my cursor when I'm circling? I don't know if you can. It's the last group there, the ASD with intellectual disability, compared to really all the other groups that stood out, that looked different, that had significant association with these inflammatory cytokines and chemokines. So that's very interesting. And there were several. It wasn't just one particular, immune marker. It was across many different of these groups, but these inflammatory ones mostly. So that was very interesting. So moving on then to looking at some of the environmental exposures, and I'll tie it into immune system in a minute. We were able to measure in the maternal blood samples, serum samples, PBDE flame retardants, PFAS, and also several organochlorine chemicals, the PCBs and organochlorine pesticides. And this has all been published. And Kristen Lyle is the first author on all these papers. And we did see some positive and significant associations with two PCB congeners in particular. Well, we saw sort of elevated risks. These are odds ratios and confidence intervals that the point estimates were elevated, but non-significantly so. For these two, for the PCB 138, 158, and 153, these were significantly elevated in the mothers of children who were subsequently diagnosed with autism. And there are several potential mechanisms or pathways that could explain this, and one of which could be immune system disruption, maternal immune system disruption. Another way we've looked at immune system is in relation to prenatal air pollution exposure. And I know this group, a lot of the people in the audience here are experts in air pollution. I'm not an expert in air pollution, but I'm lucky enough to work with people who do have that expertise. And this work was in collaboration with Dr. Heather Volk, who used to be at USC, now at Hopkins. And we were able to, she quantified prenatal air pollution exposure in all of our EMA women, and we were able to look at, we're looking at main effects, but we really were interested in also looking at the relationship between the air pollution exposure and maternal immune markers. So we had this in the same data set. And what we find here is that there are many correlations, and the red is positive correlations and then blue is negative correlations between many of these markers, immune markers and inflammatory markers in particular, and air pollution exposure. And you can see the different, we looked at NO2, ozone, PM10, and PM2.5. And the ones that are with this little caret where we do see these significant correlations between maternal immune marker levels and air pollution exposures are similar, are often overlapping between the cytokines and chemokines that we previously found associated with autism with intellectual disability. So this has been very interesting. This was published. I'm not sure if the date is 2021 or 2020. I think this is out already. Yeah, I don't think it's in press anymore. And so we're continuing to look at the air pollution data, and we've got a manuscript looking at main effects as well, which we actually didn't see. That we didn't see strong main effects of air pollution and autism in this data set, interestingly. And that's a whole nother discussion to go into, which I don't have time right now, but that hasn't been published yet. But we do see this interesting association between the immune markers and the air pollution, suggesting that, you know, the immune, that this air pollution autism or air pollution neurodevelopment association may be acting by way of altered maternal immune regulation. And we also have genetic findings. This is a little bit hard to grasp right away. And this is just a sort of a summary of what we found. We've published many papers looking at the genetics in this sample of EMA women and children, looking at both maternal genetics and child genetics. And we found, I just want you to focus on the right-hand side of this slide, where we find both maternal loci and, well, we found two maternal loci having to do with xenobiotic metabolism that were associated with the maternal PBDE and PCB levels. And we also find maternal loci that are in cytokine genes that correlate with or predict cytokine levels in mom, pregnancy samples from mom. But we also find fetal loci that are not in cytokine genes that were significantly predictive of the cytokine level in mom, which is interesting. And we also found, and so these cytokine levels here are in the newborn blood spot. We found both maternal and fetal loci that were predictive of or associated with cytokine levels in the baby. So this is all very interesting. And of course, we're working on ways of synthesizing all of this information, the immune, the environmental, and the genetic, and the child outcome, and trying to make sense of it all. And it's complicated, and the methods are are sort of beyond me, but we're working with people who are experts in mixtures and in genetics. This genetics work was done in collaboration with Dr. Lori Weiss at UCSF and Michaela, one of her postdoctoral fellows there. So one of the, so while EMA has been an incredibly rich study, and it allowed us to look at many things that weren't able to be looked at all in one study before, it had a lot of limitations. And one of the big limitations was that the data The participants in the study, all of the data in the study was based on existing information in either the vital statistics in the birth certificate files, in the DDS system files, or could be interrogated from these existing blood samples, the prenatal and the newborn blood samples. We didn't have any information on, other than the tiny bit that's recorded as part of the state prenatal and newborn screening, about, we had some demographic information. We knew the woman's weight when she gave her prenatal sample. We didn't know anything about her infection status or other metabolic conditions or other conditions that could lead to inflammation. And the only thing we knew about the children was what we could glean from the birth certificate files and the DDS regional center files. So we decided we could do something like this. Wouldn't it be great if we could do something like this within the Kaiser population, where we have extensive medical electronic health record data on both mom and baby. We have the opportunity to survey, to do additional data collection. We have great diagnostic information to determine outcomes of the kids, because these kids are getting, have been followed since birth in Kaiser, and they get diagnosed in terms of autism. We have specialty clinics that all they do is assess and evaluate children for autism spectrum disorders by experts, and so they have very good, valid diagnoses. So we wrote a grant and got funded by NIH to do what we're calling the impact study, and where we're looking at immune and metabolic markers during pregnancy and child development. So here we are. The aims are to characterize the maternal immune and metabolic profiles during pregnancy, and to determine longitudinal patterns associated with neurodevelopmental outcomes in the offspring, and to identify maternal factors, demographic, clinical, and genetic, associated with altered maternal immune or metabolic function. Both immune and immune conditions and metabolic conditions can lead to inflammation. So this is our underlying, hypothesis is about maternal inflammation during pregnancy. This is a case control study among Kaiser Permanente members. Kids are born between 2011 and 2015, so we have lots of years of follow-up on them. And this is sort of a picture of the study, just said a different way. Our main exposure is the immune profile and the metabolic profile. We're determining this both from biomarker data. What I failed to mention is that the existing resource at Kaiser I was involved with many other of my colleagues in creating a pregnancy cohort, which recruited women, just general women who were getting prenatal care at Kaiser, to participate in this biobanking project, where we asked them to complete a survey and then also donate a specimen, a blood specimen, at the time they were coming in for their routine prenatal care in the first trimester and the second trimester. So we consented over 25,000 women to participate in this pregnancy cohort biobank, and it's those women that I included in this study, and women who they were identified. It was among the women who had prenatal specimens twice during pregnancy, who then went on to deliver children who were later diagnosed with a range of neurodevelopmental disorders, who are the study participants in this study. So again, this is more of a cohort study, but we already had, the kids were already old enough to identify who had the outcomes that we were interested in. We're looking at, so we have in this study, we have the mothers and the children's medical record data, which is very extensive. We've got, we have information on sociodemographic characteristics, as well as clinical conditions before and during and after pregnancy. Many of these women also completed that pregnancy survey, which gives us a lot of information that's not captured in the medical record data. And the samples that we collected in the first and second trimester provide the ability to look at maternal genetic profiles, and then also these immune and metabolic markers, biomarkers, cytokines, chemokines, C-reactive protein, and a number of metabolic markers like leptin and other things. And we're here in this study looking at a whole range of neurodevelopmental outcomes. One group being autism, and then another group being sort of a mixture of many different outcomes. And so these are preliminary findings, have not been published yet. We're working on a manuscript where we find, so this is, we didn't have the assay data yet, but we had all the extensive medical record data, and looked at a whole range of different maternal conditions that would increase inflammation. And we found that it's in the women who had both asthma and extreme obesity in class 3, which is very heavy, that had this phenomenally high risk for having autism. And it's very, there's a large confidence interval. This is not very precise, but the bottom of this is at least a 5-fold increased risk for autism. So this is very interesting, and it makes a lot of sense when we're thinking about inflammation, and so we're looking into this further, and very eager to link in the genetic. We're currently looking at the genetic data and looking if there's a certain genetic profile that correlates with these women in particular, in terms of susceptibility, maybe genetic susceptibility. And then we're also eager to get the metabolic and immune marker data so that we can fold that in and see if we can identify a profile, a biomarker profile that might indicate increased risk. So quickly in the last part of this talk, I want to, we can't ignore the pandemic, and while we're all sort of suffering the consequences of the pandemic, it's also an incredible opportunity to do research. And since my large part of my research focus is on inflammation, we know that this is an infection that both from possibility of infection itself, but also living through a pandemic and the stress that's involved, that's also an inflammatory condition, can result in inflammation. So we are taking advantage of this unfortunate situation to look at the impact of COVID on neurodevelopmental outcome, COVID infection, and also stress-related, pandemic-related stress during pregnancy, and what that might look like in terms of risk for neurodevelopmental disorders in the children born to those women. And so we were able to get additional funding from NIH to add on to our impact study, the one I just described, to basically establish a new pregnancy cohort. These would be women who were pregnant during the pandemic in 2020, and to be able to look at this hypothesis that maternal COVID-19 infection and or pandemic-related stress during pregnancy increases risk of autism and other neurodevelopmental disorders via this mechanism of heightened maternal inflammation during pregnancy. And because we have a very large and diverse population to study, we were also able to look at disparities and racial ethnic disparities, and also in infection and stress, and also disparities in terms of pre-existing medical conditions and conditions that we know have been linked to increased risk for severe COVID, for example. And so we hypothesize also that children born to women in some of these disadvantaged groups, race ethnic groups, and also women who have existing chronic health conditions such as obesity, diabetes, or asthma will be at highest risk for delivering children with neurodevelopmental disorders. So quickly, the methods for this, it's a prospective pregnancy cohort study within the Kaiser Permanente Northern California population of pregnancies, actually of live-born children. There are many more pregnancies, but there are 45 to 50,000 live-born children every year in the KPNC population. We're studying all of these mothers and their children when they're born. We've invited all of them to complete a COVID pregnancy survey, which is based on the ECHO study COVID survey, and we've added a bunch of questions to it. To date, and this was intended, the funding was just to follow women who were pregnant in 2020, but since the pandemic is continuing, we're continuing to enroll women even now, and we will through this year for sure. We've over 22,000 women have completed the survey. The survey is self-reported COVID symptoms, exposures to COVID, or COVID exposed people. We also are asking about stress, depression, anxiety, impacts on employment, food security, behaviors that have changed because of the pandemic, such as increased TV watching or doing puzzles, or both positive and negative kind of behaviors that may have changed. Healthcare access and utilization, and now we're adding a question on the COVID vaccine as well. For a subset of these women, we are getting newborn blood spots for the babies once they're born, and these are going to be mostly to babies born to women who we know had COVID-19 during pregnancy. The study would only fund about 550, so that's why that's number, but that's what we estimate probably will be the number of women who had COVID during pregnancy. And then we'll be testing those newborn blood spots for immune markers, which will reflect maternal exposures and maternal immune system regulation or dysregulation. So we're testing for cytokines and chemokines. We're also testing for antibodies to SARS-CoV-2 and also other coronaviruses. These are our hypotheses with respect to the blood spots, that the COVID-19 infection during pregnancy will result in SARS-CoV-2 antibodies that are measurable in neonatal blood, and that risk of child neurodevelopmental disorders will correlate with antibody levels and will be increased if IgM antibodies are generated. IgM antibodies indicate a baby infection was the baby and not the mother. And that these relationships will be modified by the timing during pregnancy of COVID, and also by the maternal medical conditions and also child sex. And we have a parallel hypothesis, but this is with respect to cytokine chemokine profiles in the baby, in the newborn blood. So this is ongoing. Of course, most of the babies many of the babies haven't been born yet. We're going to be obtaining the newborn blood spots by the end of this year from the state blood spot archive, and then we'll be doing the lab work and being able to look at these questions. Just preliminary, we've got a paper under review right now that describes just some of the survey results from those 22,000. Actually, it was the first 13,000 women who completed the pregnancy survey. And we are finding in our population and our insured population, higher infection rates among African American, Latina, and Asian women, and also higher COVID rates among women with pre-existing health conditions such as obesity, asthma, and allergies. So we think that this will be very interesting to see what happens in terms of risk for autism and other neurodevelopmental disorders. And then planned future directions are to expand this pregnancy cohort yet again to include women who are currently pregnant. And if we're fortunate, we will start
All right, so it looks like Lisa is having some connection trouble, so we're going to give her a couple of minutes to rejoin the meeting and then we can continue. Hi, Lisa. You're muted.
Yeah, okay, I'm back. I got dropped for, okay, this is my last slide. I don't know if you can see it anymore, but actually, here's my last slide, which is my collaborators. I'm not sure you're seeing that.
We actually can't see it, so if you hover on the bottom of your screen and click on share screen again, the green icon.
Okay, let me see if I can do that. Oops, sorry, that was the very last slide.
And Lisa, if you don't mind going back over your last feature directions because we missed that part.
Oh, you missed that. That was okay.
Yes, thank you.
Okay. I have to, from current slide. Okay, is that working?
Yes.
Okay, so future directions, yeah. To collect, to continue the pregnancy cohort into 2021, because the pandemic is still going on, to collect pregnancy blood and stool, and this would be mid-pregnancy when the woman comes in for her prenatal visit, her second lab during prenatal care. Get the newborn blood spots for those children born to those women, run these immune panels, cytokine, chemokine panels, metabolic panel as well, and then also look for antibodies specifically to SARS-CoV-2 and hopefully other coronaviruses. And then we are very interested in linking to the air pollution databases to look at air pollution, and then also specifically examine the impact of the wildfire exposures during the pandemic, and that happened. And then we can, we'll be able to follow the kids up and hopefully in the future bring them in for other neurodevelopmental assessments or do some parent-reported measures on these kids that might pick up more subtle or some other outcomes that we can't get from our medical records. So that's planned, not funded yet, but planned. And then I just finally want to thank all my collaborators. These are just the key collaborators on EMA and IMPACT, and my lovely team at Kaiser, enjoying a cooking project that we were able to do together before the pandemic. And I hope I've left enough time for questions, and I thank you for your attention.
Well, thank you for a wide array of fascinating work that's undergoing and promises of things to come. So we'll open up the floor to questions. We have about maybe 10 minutes or so. If any of the anybody in the audience would like to ask Lisa a directed question, maybe just raise your hand or unmute yourself. Look across here and see.
Lisa, I'll ask a question. This is Rob McConnell. So, just describe again. I think it was what was it? Was asthma and obesity, or asthma and metabolic disease that had the 16-fold increased risk associated with it?
It was asthma and obesity class 3, which is the highest. I think that's over, that's a BMI over 40, I believe. So it's a very extreme obesity plus asthma. It was those women, that subset of women who had that very, very, high increased risk. We saw an increased risk for obesity itself and asthma itself, but they were much, much lower.
And did you see interactions of significant interactions of obesity and asthma at lower levels of obesity, BMIs of 30, for example? Or was it just the morbidly obese people?
It was the, well, that strong interaction was the morbidly obese. I can, oops. Oh, I don't know, I did it, what I just did. I was going to go back to my slide here. Let's see. Yeah, I don't think I have on this slide yeah, I don't have the obesity. Just there was a significant, I think the odds ratio was about 2 for asthma and any asthma plus obesity and any obesity. But then when you start looking at the level of obesity, it really shoots way up with that morbidly obese, that class 3.
And how about asthma with metabolic disease? For example, diabetes, for example. Did you?
Yeah,
is
we
it some
didn't.
specific to obesity, or is it?
Yeah, it was specific to obesity. And I'm trying to remember if we looked. There wasn't much. There was hardly any diabetes in this cohort. There was gestational diabetes, which interestingly we thought we'd find an association. I know that from Kaiser Southern California data, they found significant association with GDM, with gestational diabetes, excuse me, and autism. And in this impact study, we did not find that, surprisingly. We found an association with, I believe, with the DD, the other group, the DD group, which is not autism, but other sort of a range of other developmental disorders, but not with autism for some reason, which was a surprise, unexpected.
And how did only in Kaiser can you look at interactions of class 3 diabetes with, or class 3 obesity with anything? But how did you think to do? Was this exploratory, or was this an exploratory thing that you found, or did you have a hypothesis going into this that you would see this interaction between asthma and severe?
No, it was exploratory, and I'm trying to remember how we got there. And I should be able to answer that question, but I can't at the moment. We did not have, we had seen independent associations with, I know how we got there, because of all of the different, when we looked at the conditions individually, I was expecting to see an association with infection, with some kind of infection, which we actually didn't see. And actually that was consistent with what I reported from the SEED study, where overall, if you look at infection, you don't see any association. But when you look at specific, you might see specific infections. We also looked at fever, which is shown on this slide, and there didn't seem to be an association significant with fever. But what we did see, the only 2 conditions where there was a significant elevated risk for autism was asthma and obesity. And so I think we looked at the women, we started looking at the data, and then we said, well, what happens if you have both? Are these independent? Was the question. And it turns out, oh my God, if you have both compared to neither, you've got this whopping risk. That's how we got there.
Yeah. That's very exciting.
Yeah.
Why is this? Why do you see this?
Well, that's the big question, and we'll be able to look at, fortunately in this data set, we've got the maternal genetics, and we've got the immune markers coming, so that we'll be able to really look and see if we can find some kind of, answer that might explain it, biologic, some kind of pathology or mechanism.
Hi, Lisa.
Yes, go ahead. Go ahead, please.
Hi, Lisa. This is Annie.
Hi, Annie.
Actually, I really enjoy this and, get some picture of what you're doing. Actually, just some comment, what you see about the question about gestational diabetes. Actually, if you look at just gestational diabetes overall in what we published in 2015, the fetal cohort, we don't see that either. What happened is really diabetes either pre-existing or the gestational diabetes identified early during pregnancy, in the first and second trimester. That's having a risk. About 2/3 of gestational diabetes actually was really detected later on in the third trimester, because the screening usually happened in the end of the second and beginning of third trimester for normal risk women. In that case, actually, there's no increased risk. So when you just don't look at the timing, you don't see that one. So I think
Okay.
that's probably explain why you don't see that. If we do that one, we don't see that either. So it really happen is the early diagnosis, the one it could be those women have some beta cell defect already. They have maybe impaired glycemia or maybe pre-existing diabetes and never been detected. We know glucose fluctuated. If you never go to see a test, that you might not know you have glucose getting past that threshold, call you you being called having diabetes. So that may be the reason. Actually, there are at least two studies I know. They look at also interaction between diabetes and obesity. It's showing that there are also significant interactions with obesity. If you have both conditions, your risk is much higher. The statistically significant interaction. So now you look at asthma, obesity. Of course, asthma, obesity itself, have lots of common pathways. So it's really, I think what you say, it's kind of makes sense in a way.
Yeah. Well, I'm curious about the GDM detecting it in the first trimester, because the test, if it's undiagnosed, I mean, how do you get GDM diagnosed early on in pregnancy?
Usually they would, nowadays they could use give you a hemoglobin A1C screening or give you a 1 hour glucose screening. The alcohol recommendation is that if you have previous history of gestational diabetes, or you have older age or certain ethnic group, like in Asian, you're overweight, and then have family history, but you're don't have diabetes yet, they are screening you. You could give a 1 hour GCT, glucose challenge test. If you're above the threshold, which is 200, they don't offer, or you don't need to go to a full OGTT. Means you are, really elevated. You have to be treated right away. So,
and that's early.
Yeah, that's early. But you have normal, and then your level is standard is okay. They probably just wait until end of second trimester and beginning of third trimester, which currently is 24 to 28 weeks.
Right. Okay, that's good. Yeah, that's helpful. I can go back and look at our data and see. Yeah, I was very surprised, so I didn't look at that nuance, but because this wasn't focused on GDM. Yeah.
Go ahead, Beata.
Yeah. So Lisa, thank you very much. This was wonderful talk.
Thank you.
Yeah, and you might know that we also have been given access to about 300 autistic kids, mothers, 16 weeks pregnancy samples. They all live in the Central Valley, and we also looked at 300 typically developing kids. At least they're not in the database. So, and we ran this through a metabolomics untargeted analysis. We had actually selected these children's mothers according to high and low air pollution exposure in the Central Valley, traffic related. And what we have now 3 papers, one where we're just looking at an air pollution signal and its inflammation. Then one where we're looking at the autistic kids versus control kids under no exposure or low exposure. And there we're finding some maybe autism related pathways, including steroid pathways. And the latest paper that hasn't come out, that is actually what is so interesting with your table. We are actually comparing the mothers with high air pollution to the control mothers with high air pollution. And there we see all sorts of sugar pathways. So glycolysis and gluconeogenesis coming out. So, you have cases and control mothers, but they're all challenged by air pollution. And there's really a different pattern there of pathways metabolically already in the 16th week.
Interesting.
Yeah.
Very interesting. Okay, I'm going to look for your papers. Yeah, those 2 anyway that are out already. Yeah. So anyhow, this is great work. Thank you.
It can only be done at Kaiser. Keep going.
So we're coming to the end of our hour. It's 1 o'clock. I want to thank all of the audience. I want to especially thank Dr. Croen for taking the time to share with us some of her insights. Lots of interesting questions here that we want to follow up on. And please continue to be in touch. We will hopefully continue these discussions on either through email or continued research, collegiate research. But thank you all for attending, and thank you, Lisa Croen, for the presentation.
Thank you. Thanks very much.
Thank you.